All right, now that we've talked about how a neuron can communicate inside of itself, action potentials, now we need to talk about the next step in communication, because remember, there is a gap between neurons. They're not directly linked up with one another, so that electrical signal that it generates can't actually be sent across the gap. So what the neuron actually has to do is take that electrical information and turn it into another type of communication that can cross that gap. And since we're talking about crossing the gap, we're going to be talking about neurotransmitters. And we'll be talking a lot about neurotransmitters throughout the semester, especially when we get into talking about psychological disorders and the treatments that are, popular in current use right now that control a lot of the symptoms of mental disorders. And we'll also talk about drugs next week as well. So when we're talking about communication between neurons, we call this synaptic transmission, and it involves neurotransmitters. Okay, so if you look in this first box, you see two neurons connected to one another. Now the neuron that is sending the signal is what we call the presynaptic neuron. Okay, it is the sending neuron. It sends the information. And the second neuron, which receives the information from the first neuron, this is what we call the postsynaptic neuron, meaning it comes after that synaptic gap. Okay, so when the action potential signal travels down the length of the axon, when it finally gets to the very end, to the axon terminals, remember, in those axon terminals were those synaptic vesicles, these little pouches that contain neurotransmitters. And that electrical signal from the action potential, what it actually specifically does, it opens up some channels around here and some calcium ions come in. Basically what happens is those synaptic vesicles, they move down to the very end of that axon terminal to the membrane, and then they burst, and then they release all their neurotransmitters out into that synaptic gap. And they're just out there and they're kind of floating around until eventually they're going to bump up against the next neuron, the dendrites of the next neuron. And remember, on the dendrites of that next neuron are receptor sites. Okay, and neurotransmitters bind to those receptor sites. That's how they're able to send a signal to that next neuron. And so they bind on there very briefly, send their signal, and then are actually released back into the gap. And sometimes they float and they hit the neuron again and send another signal. And then sometimes they're actually reabsorbed back into that sending neuron to be used again for a future time. So, in talking a little bit more about neurotransmitters, since we obviously will devote a lot of time to them this semester, is that neurotransmitters, they're communicators. And some communicators, they're either excitatory and their purpose is to speed up activity in the nervous system. They make action potentials much more likely, much easier to accomplish. And so, or they can be inhibitory. They can slow down functioning or communication in the nervous system. Once a neurotransmitter has locked into a dendrite, it can either excite that neuron or inhibit the activity of that neuron, meaning they can make it either more likely that the neuron will have an action potential or less likely that the neuron can have an action potential. And if a neuron can't have an action potential, it can't communicate with other neurons. Another principle, or characteristic I should say, of neurotransmitters is this idea that neurotransmitters, they are chemical molecules. They have very chemically distinct shapes. And in order for them to actually send their signal, to actually communicate with the neuron, is they have to find a receptor site that is specifically shaped just for it. So we'll talk about, acetylcholine neurotransmitter, and it has a very specific structure and a shape that in order for it to send a signal, it needs to bind to acetylcholine receptor sites. And so it's very much kind of like we call it the lock and key principle, because like a key in a lock, a neurotransmitter must perfectly fit that receptor site on the receiving neuron for its messages to actually be communicated. And another interesting characteristic of neurons, and this is actually very important in drug research because this is one route by which drugs interfere with chemical communication in the brain, is this concept of reuptake. And you can word associate recycle with reuptake. Okay, your body was one of the original green machines on this planet. And so, as I kind of alluded to before, is that sometimes when after sending a signal, a neuron releases neurotransmitters into that synaptic gap so it can go bind to the next neuron. Well, sometimes neurotransmitters are actually reabsorbed by that sending neuron. Okay, it would be as if you just squeezed out just a little bit too much toothpaste and you kind of let go, release pressure on the tube of toothpaste, and some of it gets sucked back inside. Well, that's the concept of reuptake. And so they actually get reabsorbed by the sending neuron so they can be used again at a later date in other signals that the neuron will transmit. And as I mentioned, this is a very important route of mechanics of drugs, because a lot of drugs, for example, a lot of antidepressant medications, they interfere with the recycling process. They actually block the channels that the neuron that sent those neurotransmitters is trying to use to reabsorb those neurotransmitters. So this is reuptake.